நான் ஷேர் பண்ணுறேன் ஸ்க்ரீன் மியூட் பண்ணுவோம். ஓகே யா
researchers so our experience at that time was that was our inspiration. The number of popular so we are also one of them. So we have been to start that. We like this, we have been organizing something public level. To not get to start it.
ஜூம் ஆயிட்டு ஜூம்ல என்ன ஷேர் பண்ணல ஸ்க்ரீன் 2 yes
just follow.
yes yeah
So we have to do and various activities. We are publishing 2 magazines here. Put it, it has to product in English also. So 4 years, 6 years, we gain this just to start. Because so at that time we were very surprised to see the confidence of subsequently so every year we are conducting whenever Nobel Prize is announced. Physics, chemistry, and medicine Nobel Prize. All the topical infos we have. That all the people who are common there are leading their own experts who have worked in their own, whether it is environmental, physics or chemistry. So today also we are got a distinguished speaker going to talk of why do deadly viruses contain RNA. Very interesting topic itself. I am not a biologist, so let me not talk. Even like a brief introduction to the speaker. The speaker had that a very it's basically Dr. Arun Panchapakesan, assistant professor in laboratory manager, molecular biology laboratory, YR Gaithonde Centre for AIDS Research and Education, something named by itself, YRG CARE. Please take this call. He holds BE in Biotechnology from RV College of Engineering and a PhD in molecular biology and genetics from Jawaharlal Nehru Centre for Advanced Scientific Research. The total institute to do PhD there. Itself as a his work focuses on HIV-1 research, including viral evolution and reverse transcriptase mechanisms. With the several publications and manuscripts of them, I think I have spoken the biological term correctly. Dr. Arun is also a science journalist, contributing articles on HIV, COVID-19, the topics of the platforms like the Hindu Wire, about second year, any author magazine, Wire, Hindu, of course, I need not tell about it. He is a visiting faculty member biostatistics and amateur astronomer. And the science secretary Chandrakant. Today also he has come as an outreach program here. Unfortunately, really we are surprised here. The audience is really very small here. Normally we tend to get up. Sometimes we have enough chance. Get really basic. Continuous rain. This way, that way, able to interrupt this here. People are trying to settle in the trust. Anyhow, I just spoke to Arun that it's not an upper that counts. The quality of the audience that counts. And I know definitely quality of this. Fluent in multiple languages, he engages in citizen science and educational initiatives. And he holds a radio broadcast license, very versatile. His contributions span academic research, science communication, public education. Yeah. And let me not stand between him and you. So I welcome Dr. Arun Panchapakesan.
Thank you, sir. Thank you. I think a smaller audience always is good in the sense that you can have more discussions and it can be informal. I think not necessary, sir. Okay. My research actually spans on HIV, which is an RNA virus in itself. And you over the last few years, there will be this RNA is something that keeps coming up in the news. You got already there are 5 Nobel Prizes that have been given for RNA research. There is this year's Nobel Prize was for microRNA. You can see all of these books that the library has kept here have everything to do with RNA. And it's a very versatile molecule, actually, if you think about it. But it's a versatile molecule for therapeutics, for all other purposes. But as the molecule that carries genetic information, it's not so good. And that is where what I'm going to talk about today is actually going to be focused on, is that how is it that this group of organisms, this group of viruses really use something that is bad and survive, right? That's the essential idea what I'm trying to talk about. And please, if at any time, because we are a small number, please feel free to interrupt me and ask questions if you are stopping. If you have any doubts, I want to thank, before I start, the Tamil Nadu Science Forum, especially Dr. Tirunavakarasu and Mr. Vijayan for allowing me to speak here, and Professor Raghavendra Gadakar at IISc for giving me a lot of inputs on how life and variation and evolution and all those ideas. Okay, now that this thing's done, you might have heard all these different laws in physics and rules in chemistry. In biology, there is actually just one rule that sort of supersedes all other rules. Okay, it's this one phenomenon that underlies every single organism that you can see, right from the smallest coronavirus to the largest animal on the earth, blue whale. Everything is governed by evolution, by natural selection, right? There is nothing that does not follow this rule. Everything has to. And this evolution by natural selection occurs because of variations, because of changes. Okay, what do I mean by changes? If you look at any two individuals, if you look at any two human beings, you would see at the DNA level, we are all about 99.9% identical, right? It's that 0.1% that makes the difference in what we are, all of us. Okay, now if you were to compare this DNA similarity between human, now if you take humans as a group and you compare it with a chimpanzee, that number will be 98.8. Okay, we're that close to a chimpanzee, but the important point is that there is change, there is difference in the DNA, and for some reason we are selected and the chimpanzee is also selected evolutionarily, but we are at least slightly ahead on that line. Okay, now building on this, okay, I want to make a point that it's not always about DNA. Change does not always occur at the DNA level. The best example is this ant. Okay, this is an electron micrograph of an ant. Okay, there are 2 ants here. Okay, this is one, this is one. These 2 ants have identical DNA. Okay, the DNA is the same, but that's the full size of the worker ant, and this is the full size of the major ant. You can see that it's almost 500 times as light as the other with the same DNA. So DNA does not mean everything, and variation does not always have to occur at the DNA level. Okay. Now, what does that mean? Now, that means that how do we really know when, this variation or this change is actually important? Okay. It is only by asking the right kind of questions. Okay. Biology is the study of patterns in a lot of, change essentially. And what is the question that I am asking today? It is very simple. What have we learned in our history with disease? Okay. And to understand this, what I'm going to do is I'm going to be listing the greatest pandemics, okay, that mankind has ever encountered. Okay, I have the list, I have the name of the disease, when it affected human beings, and approximately how many people died. And I'm sure the coronavirus pandemic is fresh in everybody's mind, but in reality it's not even in the top 5 in this list. It's not the worst thing that has hit us at all. Okay, so in 10th place we have what is called the Hong Kong flu. It killed about 1 million people, 1968, 1969, that period. Then we have the Russian flu, that about 1889 to 1890, another 1 million people again. Then we had there were multiple cholera pandemics that affected us, and the 6th one was one of the more deadlier ones, 1899 to 1923. It lasted. Then there was the Asian flu, which was 57, 58, 2 million people died. Then comes our coronavirus. It's at the 6th spot. It killed about 7 million people around the world. Then we have the modern plague, 1894 to this thing, 10 million people died. Spanish flu, more famous one, 1918, 1920, 20 million people died. AIDS still going on, 39 people already lost. We don't know how many people more. The Black Plague at 2nd place, which killed 50 million people in the 1300s. This is famous. It wiped out 1/3 of the population of Europe at that time. And of course, the worst is the Plague of Justinian, it's called, which happened in the year 540. It killed about 100 million people around the world. Okay. Now, because we are all science enthusiasts, we have to really look at this table and try to see if there is a pattern that we can see here. Okay. Firstly, and most importantly, if you observe this table carefully, you will find that about half of the diseases here are caused by bacteria, right? I have highlighted them in green. 4 of these diseases are caused by bacteria over the period of human history, and the remaining are, of course, all of them are viral diseases. Okay. The other interesting thing that you'll notice is that if you look at the years, the last major bacterial pandemic happened over 100 years ago, 1923. That was the last time that there was a major problem with regard to bacteria. And the reason is very simple. It's because something happened in 1928 that really changed how we view infections. Okay, and that is the discovery of the antibiotic, Alexander Fleming, right? Everybody knows that. Now, if you look at the other half, okay, all of the ones in red are all viral diseases. Okay, there is really no medicine against a viral disease, and I'll come to why that is so in my subsequent slides. And at this point, I want to ask all of you a question. Okay, can we name some viruses? Let's start naming. Influenza, that's a good one. Oh, it's there. So, oh, you did see that. Okay. Retrovirus, that's a good one. It's a family. HIV belongs to Retroviridae. Yes. Hepatitis, which one? There's A, B, C, D, E. Which one? There's one. Actually, I'm looking for certain viruses in particular. Okay, let's take B because he said it. B is an interesting one. More. Rhinoviruses, yes. Coronavirus is there. There's Nipah, Ebola, Marburg, polio, chikungunya, dengue. All of these are viruses, right? Except for hepatitis B. Everything that we listed are all viruses that contain RNA. Okay, there's these famous viruses that I've listed, and I've divided this list into 2 groups. The only famous viruses which contain DNA that you might have heard of is hepatitis B, all the pox viruses, monkeypox, smallpox, not chickenpox. It's not a pox virus, it's actually herpes. And of course, adenoviruses, which is famous because of the coronavirus vaccine that came up, which was an adenovirus based vector. And everything on this side, all the deadly ones, the dangerous ones, the ones which we don't have cures, are all viruses that contain RNA. Now, at this point, we really have to ask the question, why are they so successful, right? I said at the beginning that they are not designed to be very successful. RNA is not a very good material for genetics. And to understand why that is, we have to really go back to molecular biology, the first class in any molecular biology lecture that people teach. What is the difference between DNA and RNA? They're both nucleic acids. They both contain strings of nucleotides, adenine, guanine, cytosine, thymine. These four are repeated. DNA contains thymine, RNA contains uracil. The other difference is, of course, DNA has deoxyribose, which is the name DNA comes from, and RNA has ribose. For almost all organisms, DNA is the genetic material. It's the source of life, and RNA just acts as a messenger, which basically takes the information in your DNA and converts it to protein, right? That's all RNA does. Now, this actually was, if you go back in history and look at how they identified, genetic material, RNA wasn't even in the picture. Nobody really ever thought that RNA could even be genetic material. They always said in the 1900s, the question was always, is it DNA or is it protein? That was the big question that people had. In fact, the earliest reference to RNA as genetic material is this letter that Harriet Efrusy Taylor, a famous French geneticist, wrote to James Watson. And she says that uh, you can see that it's written on a note from a hotel, where she says it's top secret, and she's written that in 1953, the year that actually it's dated March 14th, so that means it was 2 weeks after Watson and Crick actually discovered the DNA structure. She has written that Burnett swears from work in his lab that the flu virus has principally, maybe exclusively, RNA, and he suspects the same for polio virus. It was a big deal at that time that something like this can even exist. Okay, and I keep saying this, that it's a difficult thing. It's a very difficult to maintain RNA as genetic material. And in order to understand why, there is, I know philosophically, religiously, there may be different sort of purposes that are ascribed to life, but biologically, scientifically, there is only one purpose to life. Okay, and that is to propagate itself. Life exists in biological terms to give rise to more life. Okay, that's essentially it. That's from a biological perspective, life has only one purpose: to propagate itself. Now, when you want to propagate yourself, there is one very important thing, and there's a very beautiful example here. Okay, if you look at, everybody went through the coronavirus pandemic, right? What's the one symptom, okay, if you think about it, that is common to all respiratory viral infections? Any virus that affects your lungs will have this symptom. What is that one symptom? Throat infection, yes, but go on. A symptom? Rhinitis, yes. I'm looking for one particular symptom in common which you will have if you have a respiratory infection. Cough, right? Why is it that every single virus that affects your lungs makes you cough? Because a respiratory virus that does not make you cough will never survive. The reason is that, what is the virus trying to do? It's basically going all the way from the recess of your lungs into the air so that it can go and infect another host, right? That's the one reason why any respiratory virus that you take will have cough as a symptom. Take rabies, right? Everybody's heard of rabies. You get it through dog bite. What's the one thing that the rabies virus, I don't know if you are aware, rabies has another name. It's called hydrophobia, right? Because the patient starts fearing water. They have a fear of what's very unusual and very weird symptom, right? Why would anybody fear water? And there's a very good reason for it. Rabies spreads through the infected saliva of the animal, right? If you drink water, that saliva which has that concentrated virus is going to be diluted. It cannot spread effectively. So the virus has evolved in such a way that it will make the patient to fear water, right? It will cause that neurological symptom in your brain where you will not want to drink water, and that's the only way the virus can actually survive, right? So ultimately, everything comes down to survival. Everything comes down to propagation of life. If you want to go even further, it means you have to find a way to pass on your DNA or RNA, whatever is your genetic material, right? The same thing. I don't know if you have seen Venus flytraps, right? There are a lot of dealers in Chennai who sell these plants, incredible plants, okay? They grow in this nutrient poor soil, okay, these plants. And so they have to rely on eating insects to actually survive. But the problem is that if you keep eating insects, you will survive, but you cannot, serve the sole purpose of life. And most plants, they survive by pollination through insects, right? And if a Venus flytrap is going to eat the insect that it's coming to pollinate, it will never be able to propagate, right? And so, very beautifully, if you see the flower of a Venus flytrap, that's how it will be. The flower will be far away from the plant itself, so that it doesn't accidentally eat the insect that comes to pollinate those flowers, right? Everything ultimately comes down to propagation of life, propagation of genetic material, right? Now, having known that, RNA is a bad idea. RNA is very, very bad as a genetic material for the simple reason. If you want to propagate yourself, you have to accurately transmit that DNA or RNA. Whatever is in the DNA or RNA, the genetic material has to be accurately transmitted. Now, when it is being transmitted, there will obviously be mistakes. No system is foolproof. Even this system is not foolproof, and you have to be able to correct it, right? And this is where RNA is a bad idea. RNA is bad because if you look at these two, DNA and RNA structures, DNA, first of all, is more stable. Okay, that deoxy, the lack of that oxygen makes it more stable. That's why most organisms prefer DNA as a genetic material. Second, if you look at the structures of the two, you know that if you can see that the bases always pair with each other, right? Adenine will always pair with thymine, cytosine will always pair with guanine. And so the problem is, if one is damaged, if you lose information from one, the other strand always has, the correct its pair. So you can correct it. If there is a mistake or a problem that is made, you can always correct for mistakes, but you can't do that in RNA. Okay. The next reason, of course, is that if you see in the DNA structure, the information, these colored the colored bars are all present on the inside, so they can be protected much more easily, right, from attack or any other foreign invasion. And specified structures of DNA allow for much longer. The information can be more complex. What do I mean by this? This is the total size of, so DNA size is measured in base pairs. Okay, if you look at the total size of the genomes of these organisms, you have the T2 phage, which is about 170,000 base pairs. A bacteria will typically have 4.6 million base pairs of DNA. Fruit fly will have 130 million base pairs. Human beings have 3.2 billion, and a canopy plant has 150 billion. And it's only because all of these organisms have DNA as genetic material, and that number is not small. A billion. The issue with that word, a billion, is that we keep using it without realizing exactly how big that number is. And just to illustrate, okay, as to how big our DNA is, I'm going to give you a small example, right? We usually, a billion is 1 followed by 9 zeros. Okay, it's 10 to the power of 9. That's how you write that. Okay, and in most contexts, we write this word billion with these guys, right? We associate it with people like these rich billionaires, right? But to really understand how much money how big a number that is, I'm going to give you an example. Okay, imagine that I'm giving you 1 lakh rupees a day, every day. Okay, I give you 1 lakh rupees a day. You work 7 days a week, 365 days a year. You don't spend any money. Okay, and you work like this for 2,000 years. Okay, so it's an absurd number, right? 1 lakh a day is a crazy salary. 30 lakhs per month, 7 days a week, 365 days a year. You don't spend. At the end of 2,000 years, with that kind of salary, you will have $1 billion, right? That's how big that number is. And of course, this guy, if you want as much money as him, you have to work at that salary for about 9 lakh years, because he has $460 billion worth of wealth. Okay, it's obscene. The point I'm trying to make is, a billion is huge. If I start counting from 1 to 1 billion in seconds, if each second I count one number, then I reach a billion in 31 years. That's how long that is. That's how much information is present in every cell of our body. Okay, but so now this has this much information. Each time a cell divides, okay, that much, that 3.3 billion base pairs has to be accurately copied. Okay, and the enzymes that do this job, they are called polymerases. Okay, these enzymes, it's their job to copy. Each time a cell divides, they'll copy that 3 billion, they'll make it 6 billion, and then one copy goes to one cell, the other copy goes to other cell. Okay, the question is, this information is not always accurate. Okay, DNA, the enzymes that copy DNA, they make a mistake about once every 1 million base pairs. Okay, but the ones that copy RNA, okay, they cannot have this error correction property because of why I already explained. And so RNA makes mistakes once every 10,000 base pairs. Now, the problem is you can't make too many mistakes. Okay, I said that the difference between human and chimp is 99. I mean, two different humans is 99.9. That arose because of these mistakes that the DNA polymerase makes. And that's 1 in a million base. Of course, there are other ways to correct these mistakes, but 1 in 10,000 is just too much, right? It's not possible. You'll obviously end up making a mistake that will kill you. So how can RNA viruses really exist? Okay, that's if you look at it from that perspective, the first thing that we need to understand is, what does a virus really do? Okay, it has only one purpose. Like I said, like all other organisms, it also has only one purpose. It will infect your cell, it will make copies of its genetic material, and it will go and infect a new host. That's it. Outside the host, the virus is actually dead. Okay, why is that important? Because it means that now we have a way to actually eradicate viruses. We can never eradicate bacterial diseases for this reason, because inside a host, if you have a bacterial disease and there's no vaccine available, let's say, out of 5 people, let's say 3 people get the disease. But when you do give a vaccine, everybody is immune. Okay, you stop giving vaccination, the disease will come back because the bacteria is alive outside the host also, right? But that's not the case with viral diseases. If you give if a virus has no vaccine, if you vaccinate and everybody is clean, the virus cannot replicate, right? And the virus is eradicated. That's exact. You can see that we did that with smallpox. We have almost done that with polio and measles, mumps, rubella. All of these are all viral diseases that are on the verge of being eradicated. And this is why you can eradicate viral diseases, because outside the host, the virus is dead. It needs a host for survival. Okay. So knowing all of this, now if RNA viruses, the copying mechanism is so many mistakes it makes, it's unstable and all of that, the transmission and survival appears impossible. Okay. Oh, by the way, there's one important thing that I forgot to tell, which is that the transmission of a virus is the most important part of its life. And if you really look at it, viruses that transmit quickly, like through the air, are those that kill you quickly, right? Take Ebola. Ebola will kill you within three weeks if it is going to kill. Coronavirus, forget Ebola, right? If you were going to be affected and if it was going to kill you, the average time the coronavirus took to kill people was between 2 to 3 weeks. It has to transmit to a new host before it kills the existing host. Otherwise, the virus will not survive, right? And that property holds for every virus. Look at viruses like HIV and human papilloma. These viruses take time to kill. They are not airborne. They have the time. They can, they have easy route. They have different, more sophisticated modes of transmission. But viruses like any respiratory virus, they have to transmit. A virus in reality does not want to kill you. If a virus kills you, it's its own death sentence, because it cannot survive without you. It has to go to a new host, right? Anyway, so if the reason RNA viruses are so dangerous is because they don't exist in one form. Why? Because I said that the copying mechanism makes mistakes. So let's say that this is the original virus that infected you. When that virus is being copied inside your cells into other forms, it will make 3 mistakes, it will become new forms that will go and infect more cells, you'll have more mistakes, and the more, so on and so forth, right? You might have, this is the reason why in the coronavirus pandemic, one of the things that you would have heard was all these variants that kept coming in. You had, first the alpha, then you had Omicron, then Delta. All of these rose because of this. When the virus was doing this, one of these, let's say this one, would have been more deadlier than the one that is originally infecting, and that's the one that was successful in the population. And So this ability to make mistakes, it's called quasi species. Okay, it's some viruses do it much better than other viruses. HIV, for example. So HIV does it at such an amazing rate that if you really look at all of the influenza and coronaviral variants, okay, all of them in the world, in every patient in the world you take and you count them, there will be more variants in one HIV patient's body of HIV. That's how bad HIV is in case of this particular this thing. Okay. The second thing, there are basically three things that happen because of this RNA polymerase making mistakes. Okay, one is this, that there are multiple forms. So your immune system really is going to struggle. The immune system, it doesn't know which one to fight. It's going to try to mount an attack against all of them, and you get effects like, your cytokine storm or all of those effects, which are really the immune system struggling to fight this many different escapes. Okay. The second problem that happens is that RNA viruses have very small genomes. What do I mean by, like I said that humans have billions, RNA viruses cannot afford, because if it is too long and it makes one mistake every 10,000 base pairs, it will make too many mistakes for it to survive, right? Now, if you look at a graph, if you make a graph of size of the total DNA, total genetic material versus how many mistakes does this polymerase make, mutation rate, you will find that the larger animals, higher organisms, humans, all of these bigger animals, they'll have big genome sizes with less mutation rate. Okay, they have to. Otherwise, because let's take, for example, a mistake in, let's say, an important gene like insulin is going to be lethal if you cannot survive without things like insulin. Okay. For bacteria, the genome size is smaller, the mutation rate goes up slightly. DNA viruses, they are going to be somewhere here. The genome is even smaller, but RNA viruses are at they have a very high mutation rate, and because of that, the size at which they exist is much smaller. Okay. And if you look at the sizes of genomes, the DNA viruses are generally about 1,000,000 base pairs. That's the average size of the genome. RNA viruses are just 9,000. They're very small, very compactly packed genomes they have. Why is that bad for us? Because if the genome is small, it takes less time to copy, right? In the same time that the DNA virus will make 10 copies, an RNA virus can make 100 copies, right? Because it's 10 times. You get higher viral numbers in a short time. Once again, your immune system is not going to be able to handle it. If you make too many viral copies in too short a time, it's going to be too much for the immune system to actually handle. And lastly, most importantly, zoonosis. Okay, this is RNA viruses are all 90% of all RNA viruses come from animals. And increasingly, as we keep, going into forests, cutting down trees, and make more and more of these incursions into the wild, we are going to see that more and more pathogens keep coming. You name the virus, coronavirus, we know it came from bats. Rabies comes from bats. HIV came from monkeys. Almost all viruses that contain RNA would have come from this thing. And the reason is very simple. Viruses are usually very host specific. What do I mean by that? Is that if I take a plant virus and I eat it, nothing is going to happen to me, because it's a plant virus, it's not going to infect me. But that's not the case with animal viruses, because I said that a human and a chimpanzee is 98.2% identical, right? So that means that any one of these variants in a virus, let's say I take any one variant, which could be very good at infecting humans, might be present in that chimpanzee or whatever other animal from which it came, and it can easily jump. Because it makes this many changes, all it takes is for one, mutation to be good, and that virus is now going to proliferate. And that we see that very beautifully with HIV. We know that HIV actually jumped species 4 different times. There were 4 times that HIV came from monkey to humans. I'm talking about the dominant group. HIV basically came from 2 different monkeys. We have chimpanzee and Suti Mangabe. They're both monkeys in Africa. And the hunters there in Africa, they consume what is called bush meat, which is chimpanzee meat. And all it took was, there was a related virus called SIV, simian immunodeficiency virus, which jumped species. And we can, by genetic evidence, we know that this jump from chimpanzee to humans occurred 4 different times. We can tell this, that it came as a result of this zoonotic transfer from monkey to humans. Every year, Okay, every year we find 2 to 3 new RNA viruses that can infect humans. They're discovered, and any one of them could cause the kind of pandemic that we faced with SARS-CoV-2. Okay, and like I said, RNA viruses, they violate a very fundamental rule of life. Okay, I showed this. Every organism, okay, has what is called an error threshold. What does that mean? That means if the mutation rate goes above that, that organism cannot survive. Like if the mistakes that are made by the polymerase cross this value, this group of organisms cannot survive in nature, right? And usually this error threshold is far away from, in this graph, the error threshold is far away from where the organism itself is. You can see that for bacteria, that error threshold is slightly higher because the genome size is smaller. But in each case, as you move, the error threshold goes up, except for RNA viruses, where that error threshold is very, very close to where they actually survive. Okay. And I think this is one of the most beautiful tenets of evolution, because every law, every rule that we know says that it is not possible for an organism like an RNA virus to even survive because of the mistakes, because of the way it lives. But I think it's the fact that they exist is proof of the most beautiful, that they are one of the most beautiful products of evolution. A lot of people ask me, but do you consider viruses to be living or dead because of the way they live? In reality, I think RNA viruses are one of the most beautiful products of evolution, and the reason is because they still don't, while they violate everything else that is known about life, they still don't violate the fundamental principle of life itself, which is to propagate yourself. They found a way, and they ensured that their survival is always maintained, right? That's all I have. Thank you so much. I'd be happy to take any questions that you would be having. Yeah. 45.
Good lecture. Very informative. Thank you.
Thank you, sir.
I think it's an excellent demonstration of how to use slides. Very effective. Thank you.
Any questions?
Can I ask question through?
In terms of the
Meeting?
Yes.
Because RNA is a survival rate in terms of the error mutation. RNA has got a very less survival chances compared to other things. Yeah.
Exactly. It looks like that, but in reality, they operate very, very close to that error. So if the major problem with RNA viruses is that for us, if you look at the number of progenies that we have, it's one, let's say child per pregnancy, right? For human beings. For animals, it's slightly higher. Each time an RNA virus infects, it can make up to 1,500 different or 2,000 different viral molecules. Many of them will be defective. All it takes is for one to be good. So that's how they survive, by numbers and this thing. So even though the error threshold is very, very close to where they survive, they can still survive by sheer numbers. That's how they work. Yeah. Statistically, they will be more successful. Yeah. I think we had a question there.
I just want to ask, why is it called mistakes? Because that is really also helping them survive. I mean, there's a mutation.
So how we refer to it in scientific terms is if whatever is the baseline, we call it wild type. Anything that changes is going to be a mutation. It's going to be a mistake as far as that is concerned. See, evolution works only on the basis of natural selection. There are many genes that we have today which are important for us, but in other contexts they may be a mistake. For example, the best example I can give you is there's a mutation in a gene which makes people in Africa, they have this mutation which makes them resistant to malaria, because malaria is really a big problem in that region, right? But this small group of people, they have a mutation in a gene that makes them resistant to malaria, but that same mutation causes sickle cell anemia. So it's really the context of what you are looking at that you can call it a mistake or not. If there is a change, if everything has, let's say, one given base pair at that position, and there is a change to that, we call it a mistake as far as that context is concerned. If it helps it survive, yeah, I mean, a mutation would be the technically correct term. It's not a mistake per se, but from the perspective of the polymerase that is copying it, it's a mistake. That's what I meant.
Can a virus can be treated as a single pathogen?
So the problem with viruses is that you can't really treat it as a separate pathogen, because viruses are made of the same thing that your cells are made of. If you cannot, a bacteria is different entirely. If you give an antibiotic is only going to go and hit the bacteria, but a virus doesn't work like that. A virus is made of the same membrane, everything. It uses your own cells. So anything that you give to target the virus will also target your own cells. So as a single pathogen, I think it's difficult to classify it as one pathogen. It's like a big group.
Because it is causing so many symptoms of its infection.
Yes, that's different viruses. All of those symptoms are really, it's an evolutionary race for the virus that the virus tries to transmit. That's all. So the different viruses will cause different symptoms based on what they want to achieve.
Protein synthesis. Then I have one
How
question.
it is different
in
every individual?
For virus, please.
So I'll just come back to you, sir.
How
Yes.
it is different in every individual? Because every individual it has to have their own specific RNA.
Okay. Okay. So you're asking, why are the symptoms different? Why is the severity different? There are multiple reasons for that. We start off, one reason is the immune system itself. For example, prior exposure, the immune system works by, remembering pathogens. If I have been infected with a similar virus, like for example, take the vaccines, the smallpox vaccine, the first one was actually made out of cowpox. If you have been infected with cowpox, your body will remember that and it will, protect you. So if I have been exposed to a pathogen and you haven't, and the same virus infects both of us, I will be able to deal with it better than you. That's why responses differ. That's one reason. The second reason is there's also something called how much is the initial dosage. Let's say that the starting amount of virus in your body, what started the infection was 2 or 3 viral particles, whereas in my case it was 50. Then the progression will be much faster in my case than in yours. So there are a lot of different factors why that happens. Yes, sir.
No, it's not basically because the virus vaccination is done here long after that virus is posted. Then the effort to produce that vaccination more or less becomes irrelevant after some time. In the sense here, it operates only that particular time. After that, it becomes irrelevant.
Yeah. So, but
Is it
then
like that, or?
it is like that.
Huh?
So I don't know if anybody born after 1986 was not vaccinated for smallpox, right? I did not receive the smallpox vaccine. Many of the people here would not have received the smallpox vaccine because by that time smallpox was gone, right? There was no smallpox left in the world for you to vaccinate. Can it come back? Yes, it can. How? Zoonotic transmission again. If we have monkeypox, right? It's circulating in Africa and all. The smallpox vaccine, whoever received it, gives you to an extent protection against monkeypox. Not a lot, but if it becomes a big problem again, monkeypox, let's say it starts spreading, creates a big problem, the government will restart vaccinations. You will restart and keep this thing.
I got 100 years or something like that. So totally it would have mutated in a different.
Could be, could not be. We really don't know. There is a very good chance that what smallpox will come now will be very different. It will not be the one that was circulating earlier. That is for sure. There will be a lot of changes, but it's unlikely because smallpox itself has been known to us since 2000 years. So for 2000 years, it has been changing. And we sort of identified it, classified it only around 1850s or something that time. So, I mean, Jenner's time actually, 1750s. So, last 300 years, we know, okay, with smallpox, we study it and all that. But it has been circulating for over 2000 years. So, it will come back. I'm sure it will come back in some form someday. But we already have the vaccines ready. It's much easier to make vaccines against DNA viruses. So, we are safe. We are not that worried.
The most successful virus is rhinovirus, right?
One of the most successful.
one of the
yeah.
most successful virus. So won't the highly pathogenic virus won't the mutation eventually be naturally selected to become a common cell or a very mild pathogenic virus
Yeah, that's
to
an
become a
interesting question. I don't know of any commensal virus. And the reason is,
now
okay, so
commensal but
his
less
question is
pathogenic?
interesting. A commensal is something that lives with you and helps you, like lactobacillus, for example. The curd is basically just bacteria, right? The thing is, if you eat curd, it goes into your gut and it helps you digest your food, and it takes, whatever nutrition you eat, it also takes a little bit. That's a commensal. Um, so some organisms have been known to live in harmony with viruses. Bats, for example, that's why there are such brilliant reservoirs of you name the virus, most of them would have come from bats. SARS-CoV-2 came, rabies, Nipah, Ebola, all of these are, the reason is the bat's immune system has found a way to live in balance. The virus does not cause that much symptom in bats, but it allows low level, it will keep replicating until such time that it can transmit to a host where it will be replicating in big numbers. Same case with SIV, simian immunodeficiency virus. It doesn't cause symptoms in the monkey, but when it transmits to human and becomes HIV, then you have a lot of symptoms and other problems. So yeah.
So eventually
Can I ask one
the
question from remote?
virus can become like less pathogenic in
It
nature.
can. You have to leave it for really long time, and it usually, this phenomenon what you are describing usually happens with viruses that take a long time to cause severe problems, like HIV and other viruses, which take time to. If a virus kills you too quickly, there's really no time for that kind of evolution to happen. So yeah.
One question from YouTube.
Oh, is it?
Yeah, so people who are watching it live stream, one of them has asked a question, Mr. Aditya Verma. So he's saying, thank you very much for the very nice lecture. You mentioned that RNA virus is not interested in killing the host. Rather, it wanted its host to survive for the purpose of propagation.
Yes.
Then what is the reason for so many casualties in pandemic like
Yeah,
COVID?
that's a very good question. The death of the host is an unfortunate consequence that is bad for both the host and the virus. In most cases, the death occurs because of the host's response to what the virus is doing. Yes, the way the virus sort of survives is by hijacking your cellular machinery. And when it does that, it's going to have an effect on the host. Now, really, it's a balance between, in most cases, viruses are designed to transmit before one of two things happen. Either the host dies or the host's immune system clears the virus. The virus has to transmit before either of those two things happen. The virus is just doing what all other life forms do, propagate itself. That's all it is designed to do. The death of the host is something that the virus also doesn't desire, the host also doesn't desire. It's an unfortunate consequence. It's at the very border of living and non-living. Inside the host, it's living. Yes. But it's only alive inside the host. Outside the host, it's a dead entity. I can take coronavirus and put it in plants, nothing will happen. It has to have a specific animal host for it to survive. That's why it's, but that's not a bacteria. Bacteria, if I put it may not survive, but there are always bacteria that can live even there. Yes.
Yeah. So vaccines work by actually antibodies getting created in our
Yes.
body in order to fight for a future attack,
Yes.
right? Now, when you talk about RNA viruses, it keeps on mutating. So
Yes.
if I'm going to prepare myself with some antibody now, it's not going to be effective later on, right? Because it would have evolved a lot.
That's true, partly. You might have read reports during coronavirus also that the vaccine is not as effective against Delta. It's not as effective. That's the reason, because there is, see, the problem with, especially with coronavirus, what happened was we artificially selected for the variants that came later. If you make a vaccine that is so strong against one variant, okay, let's say Delta and the first one, Alpha, and then there's this Omicron, and everybody takes the vaccine, okay. Now, one of us, let's say I have that, in my body that Omicron variant was generated. That's the only variant that can infect everybody because it's unique. So you created that survival mechanism for Omicron to propagate. right? The natural selection happened. In this case, the selection was that vaccine that I gave. So you're right. In many cases, antibodies will not be as effective, but the body itself makes antibodies with a very similar mechanism to how viruses evolve. If a virus mutates, the antibody will also mutate accordingly to, fight the virus. The problem is, in this case, the virus is 2 steps ahead.
Yes.
By the time the antibody catches up, the virus is already ahead. That's why it's not just antibodies. There are other arms of the immune system that help. In viruses like HIV, you can't do anything because what you said, it's a retrovirus. You said retrovirus, right?
Yes.
Initially, yeah. So it's a retrovirus. So HIV, really what it does is it takes its DNA and mixes it with your DNA. So there is no way for it, the only way to get rid of it is to take that infected cell and throw it out, which is very difficult, especially because the virus sleeps. It doesn't always produce new viruses. So that's a problem with HIV. But yes, your question is valid that antibody responses usually are this thing, it takes time to evolve.
Thank you. Any questions, sir? It vaccinate against certain diseases like monkeypox. Does it make us vulnerable to some other disease?
No. To a disease, no. See, in most cases, vaccines are, especially in olden times when the polio vaccine especially was new, what they were giving was basically a weakened version of polio itself. In that case, yes, sometimes the vaccine itself may cause mild effects of polio, but for the most part, vaccination against one will not make you susceptible to any other infection. I know that there are a lot of these, especially with the coronavirus vaccine, there was a big hue and cry that, there are side effects and all of that. The truth of the matter is every vaccine will have a side effect. We just realized it because coronavirus it's like this thing that affected almost everybody. So we were all more observant for it. But a vaccine generally will not make you susceptible to any other disease that way.
So, and then when it comes to this COVID-19 virus, after that, Bill Gates has said that, in the future, we'll see several such pandemics in the world.
Yeah.
So what's your view on that? And also, when it comes to COVID-19, mRNA vaccines developed by companies like Pfizer, Moderna,
yes,
played a major role. So what will be the role of mRNA vaccines in fighting future viruses?
Okay. So the answer to your first question, if you look back at history, okay, for the last 500 years, okay, for which we have records, there have only been 4 years where there is not one single pandemic circulating in the world which has not killed at least 10,000 people. Like, pandemics are all the time. I mean, monkeypox is going on right now in Africa. They are always happening. They will always be, as long as there is life, there will always be viral diseases that will be spreading, right? And in fact, the trend going viral comes from the way viruses spread, right, so quickly. It will always happen, and there will be, history is proof. I showed that initially, right? The top 10 pandemics is proof that they will keep happening. Every 10 years or so, there will be one major pandemic that has happened. This decade was the coronavirus. We had swine flu before this. We had, I mean, in India, we are generally less, sort of scientific research and epidemiological research is not as great as it is in the rest of the world. So we are not as aware of, unless something major like the coronavirus comes up, we are not very, great at identifying and classifying viruses, but pandemics happen all the time. Right now, there is monkeypox going on. There may be other smaller pandemics. Coronavirus is still present in certain pockets in the world. HIV is ongoing. It's been going on for the last 39 years. Even though new infections are going down, it's still going on. And as far as mRNA vaccines are concerned, it's really a revolutionary technology. It will play a very major role in a lot of future diseases. But so there's no one solution to any, like, you can't say that mRNA vaccine is like the answer to every single disease there is. mRNA vaccines are useless against HIV, for example. The reason I just said that it's inside you, that it's part of you. So unless, it's definitely an important technology, it's amazing, and it will definitely help a lot of diseases that are going to come up, especially respiratory viruses, where you can quickly sort of make the vaccine. If you recall, the mRNA vaccine coronavirus was identified in November 2019. By November 2020, the vaccine was ready. So it was that quick that you can make it. And that's the really big advantage of mRNA vaccines, that you can make them very quickly, very little safety tests and all of that you can do. Yes. Yes.
Russia recently announced that it has made an mRNA vaccine for all cancers. Is that very credible? That they're able to make that in Russia?
I think Britain announced one. I'm not Russia. So, okay, look, cancer is a difficult thing. Cancer is really not an infectious disease. Cancer is your own body basically going, the instructions are going on uncontrolled cell division, right? There are other technologies. I don't know about this particular vaccine that you're talking about, but there are other technologies for cancer that are really coming up now. CAR T cells is one, immunotherapy is one, and we have a sufficiently good success rate with chemo and radiation so far. But I don't believe that there is a, if that announcement, I haven't heard of this, but I don't. Oh, I wouldn't trust too much that Putin says.
We always have parallels, like same research going on in other countries.
Yeah. Yes.
So is there any possibility that
Absolutely. We Indian researchers are they're doing everything at par with the rest of the world. The real difference between Indian and the rest of the world is that the numbers are much lesser in India. The number of top class research institutes, research laboratories in India are less, whereas they are higher in the places like US and all. But the places which are doing the top class research are looking for vaccines, are looking for other these things. The only unfortunate thing about Indian vaccines so far is that we have never really made a vaccine that's really made in India in its true sense. Our Prime Minister said that the vaccines are made in India, but in reality they're only manufactured in India. The technology was never ours. The Serum Institute was Oxford's technology, right? What is that? Covaxin was this guy's, the Covishield. Covishield was not our technology. Covaxin was also not ours. It was bought from some other country's technology, and we just manufactured it in India.
First, that was from biotech. Covaxin is ours.
Covaxin is Bharat Biotech, but even he did not develop the vaccine himself. Krishna Yella did not develop it himself. It was, the technology was somewhere else. They took it and they made it, and they did a lot of safety studies and all.
It's called made in India then.
It's not. It's manufactured in India. That's the only difference. We
So is the Covishield.
Covishield also, yes. Covishield is actually a very good story. He was, so in the interim period when we had this lockdown and everything, Poonawalla realized that he was the only person in the world who could really supply and make Covishield at the, so that lockdown period, he really spent building infrastructure for, the manufacturing those vaccines at that scale. So he's like the ultimate businessman. So he knows what he was doing. So yeah. Yeah.
So, like, if in the future, if there are lots such pandemics like COVID-19, what would be the triggers which would cause that, sir?
Man-animal conflict. That's a very straightforward answer. Viruses you will only encounter these new viruses if you go into places that you haven't gone before. Leave the forests alone, leave those animals alone. Don't go cutting into forest and, looking for more urbanization and other things. That's the simple answer to that problem, actually. I said 90% of all RNA viruses that come up are a result, actually, RNA viruses, even DNA viruses, they come from man-animal conflict only. Most of these viruses come because we go and, into the forests and all of these places where these viruses are there in plenty. And
so climate change will be a trigger, sir. Like,
climate change will be a trigger because in some sense, everything exists together in a very delicate balance. If your climate change causes, like the death of some species or some fauna, naturally there will be a change in the ecological balance, that those, the entire cycle will be affected. There is a possibility that these animals come out of wherever forests or wherever they're living. If the forests are going down and shrinking, everything is connected. Rain cycle pattern is one important reason why forests may not thrive. Forests may not thrive. The animals might, come into other places, like take bats, for example. They are the biggest problem right now. If let's say their natural habitat is declining, they'll go and find other spaces like buildings and all of these places. They'll come and this thing, they'll, you know, I mean, we say sort of, they are coming to our space, whereas in reality, it's the other way around. So, yeah.
So World Health Organization has declared that like antibacterial resistance is a like silent pandemic or something like that. So even when it comes to COVID-19, there was many reports that many people died because they didn't have resistance towards antibiotics or something like that. So like how do they, like you said that virus and bacterial infection are like separate things, but how do they relate it and fight future pandemics?
So antibiotic will do nothing for a viral infection. It will not even, it's as good as taking nothing. The reason your doctors give you antibiotics when you have a viral infection is because your throat and all of these regions are inflamed, and that makes it an easy target for bacteria to come and cause secondary infections. So when you take antibiotics, ideally, look, it's a problem because scientists are, of course, coming up with newer and newer antibiotics, but the older ones, because of indiscriminate use, the problem is both with the doctor and the patient. Doctors ideally should not indiscriminately give antibiotics without checking what is the underlying cause. Patients, if you are given an antibiotic, take the full course. If a doctor tells you take it for 5 days and you take it for 3 days and then you feel better and you stop, that's where that antibiotic resistance is going to develop because some bacteria may not have died completely. It may have, when it changes, these mutations that it will make will evolve and it will generate antibiotic resistance. There is no such, sort of correlation between antibiotic resistance and viral infections, because viruses they don't respond to antibiotics at all. Today, the biggest problem in India, especially with regard to tuberculosis, is drug-resistant tuberculosis for this very reason. Because tuberculosis is a very slow-growing bacterium. If you're given antibiotics, you have to take it for the full course. If you don't do it, the bacteria is going to develop resistance, and then that's the one that's going to transmit. So everybody will soon have drug-resistant pathogens there. So that's a global problem actually. You are right. But it has nothing to do with viruses.
So my final question is like, what will be the role of AI in preventing these things in the future?
AI actually has a very important role because so the primary reason why AI is right now being considered is that AI can do things that normally we would take years to do. For example, let's say there are all of these chemical libraries, FDA approved libraries that ideally need to be tested. Some molecules in those libraries may actually be good candidates for drug targets against bacteria or viruses. But if I have to do it, I have to sit and manually write a grant, go to the government, ask for money, build the infrastructure, buy that library, test each compound, take cells, put virus, check each compound. But if the structure of the virus is proteins and the drug is available, AI can actually shorten that. I don't have to screen hundreds and identify 10. AI can directly tell me that it looks like from that 100, these 20 are likely to be candidates. So it really cuts short the, sort of screening time for drugs. It cuts short, the way these drugs will interact with proteins. So all of those things, definitely the future is looking bright with the use of AI in diagnostics and medicine. But we really have to see how long it takes. Right now, AI access is it's a very select group of people who can actually harness AI to do, medical research. It has to be more accessible to other people for that to happen, actually.
So a lot of people say that, India is a good destination for clinical trials. So
it's the ultimate destination.
Yeah. So like, say, for example, India has different demographics and United States has different things.
Yes.
Their food habits, everything is different.
Yes.
So how does like making clinical trials in India help us manufacture vaccines or medicines, drugs across the world?
So that's a good question. Usually, diet and all of these factors have less to do with vaccine responses and all. Usually, okay, in most cases, it's really the response to a vaccine is the same, because all of us are human. More response will be more or less the same. The real challenge is when it comes to when you're really looking at genetic diseases or something that has a lifestyle component to the disease itself. There, yes, you cannot have, like, for example, if you're targeting something that is going to prevent diabetes or something like that, there you have a lifestyle component that comes in that case. Yeah, using India as a sort of drug resistance, like as a lab rat in some sense, is not a great idea. We are trying very hard, especially scientists in India, to try to prevent this from the rest of the world from looking at India just like a hub for clinical trials. It's not right. Our own scientists have to be doing. We are doing it, but there are. I mean, I have been lucky to work with two big organizations who actually have access to a lot of these samples. But for the most part, Indian the scientific infrastructure really needs to be better, because one thing in India is that we really have to come up with a more ethical there's something called an ethics board and all of that when you want to do human trials. We don't have a very good system for all of that yet. That has to be brought in, and of course it's happening slowly, but it'll take some more time for it to.
And little bit deviated topic, sir. I spoke with a medical professional. He said that fermentation will be the future of medicine manufacturing or something like that. So, like, do you have any opinion on that?
Look, even right now, a lot of anything that is biologically made in the absence of air is fermentation, basically, right? I mean, it's not just beer and the other things, that alcohol that is fermented. A lot of antibiotics are secondary metabolites. A lot of them come from this thing. So, I mean, it's a generic statement, but it's not really true. There are other ways to make this thing. If it's a biological product that comes from an organism, yeah, then fermentation is one way to produce it.
Thank you, sir.
He said somebody told him that fermentation will. He said fermentation, right? Oh, do you mean probiotics? Yeah, probiotics, of course. Gut is, we all know that microbiome really has, we are slowly beginning to realize that the bacteria in your gut have a very, very big role to play in health and disease. And it comes up in like unheard of places. Alzheimer's, for example, nobody really thought that it can be affected by the bacteria in gut, but they did an experiment. They took bacteria from the gut of one mouse, put it in the other, and Alzheimer's appears to be related to that. So we are still working on that. So in that sense, yeah, probiotics will have a important role, but it's still under research.
Following up on the question of AI, researchers in America, they have access to digital twins of our bodies. Is that something? Digital twins. They reconstruct a human body with all the parameters, and they will have access to that digital clones as a target demographic, and they'll be able to test all these research parameters. So, is that something an Indian researcher will come across?
Okay.
Have you come across something like that in your?
As of right now, it's impossible to digitally create a human body with all its complexities.
Okay.
It's not possible. It's just it's there's too much complexity. There's too much variation among individual humans for that to happen. Maybe 20-30 years into the future, that maybe it's that far away.
It was actually started back in 1990s, I guess.
You can always create the models, but it will not be a full representative.
It was used to treat a congenital heart disease for a person, and she's now a medical professional, and she's into pediatrics and sort. But it was started late back in the 90s, so that is why I was wondering if you come across something like that in India.
It's not happening right now. It's not possible to predict what would be a person's response based on digital information yet. Maybe we'll get there someday. Not right now.
Okay.
So, and I recently attended a seminar by Plant-Based Food Association of India. So what they said is something like, this vegan meat, lab meat or something like that, and plant-based meat and things like that are much more effective than normal animal-based meat.
Yeah.
And there is certain challenges, like there isn't much of fermentation infrastructure to manufacture all of these things. And one expert suggested that, like there are a lot of distilleries and things like that, and they have fermentation facilities, but a lot of them aren't fully utilized. And these facilities can be utilized for this type of things. Is it something like whatever involves fermentation, the facilities can be used interchanged manner or something like that?
You can repurpose it. It won't be the same facility. If it's the same organism that is doing the fermentation process, then you can use it. But in most cases, it's not the same facility. Look, if there is a market, the industry will find a way. That's usually the way to go about. The way scientific and any technology works as such is, if something is impossible today, it's merely expensive tomorrow, and then routine the day after tomorrow. So that essentially follows the trend. At one point, DNA sequencing was considered a novelty in the 80s. If you can do it, it's a crazy thing, Nobel Prize research. Then it became routine. Today, you can go get your DNA sequenced. You can know all 3.3 billion base pairs of your in a file. You can keep it. So that's how it is. If you can repurpose it, you can create it. But as far as vegan meat and all these things are concerned, yes, it's true that, see, what most people fail to realize is that in this whole debate of vegetarian versus non-vegetarian, there are some people who take ethical and moral high grounds with saying that, it's better to be vegetarian, it's better to be non-vegetarian. The fact of the matter is that in the course of human evolution, we have been grown, and we have the last 2000 years is what we have had proper civilization and all of that. Before that, what we were all, for the few million years that we evolved as human beings, we have been meat eaters, we have been fruit eaters also. It really depends on what was available at that point of time. Now with all of this, it really comes down to personal choice. If you want to eat, you eat. Nobody's, that's the end of the story. The rather bigger problem is that there is a entire section of, especially India, who don't get to eat at all. So that's the bigger challenge than this. India, I don't anticipate a market for that. Maybe among the super rich, but not the common man.
Yeah, I was asking with respect to climate change, sir. Like I read a lot of reports which says that dairy farming creates major factor for
it's true. What you're saying is true. That the meat industry does contribute to climate change, but there is also the fact that if you remove it, then you're also taking out most of the world's, like more than half the world's food supply. So it's not that simple to really change it, and maybe gradually things may change. It's difficult to comment right now because there are a lot of cultural and social factors also associated with the question that you're asking. It's not purely scientific because meat eating, like I said, is also a matter of personal choice. So we don't know. It's difficult to.
Any questions? Okay.
There's a home-based kit of DNA sequencing. Have you heard about that? Like it's available in markets.
Home-based?
Yeah. It's the size of a hard disk and comes to around $2,000 to $2,500 if I understand properly.
$2,500.
I don't think Reliance in India. $2,500.
Oh.
And is there a business aspect to how much research industry can develop?
Look, I don't.
No, like bringing in investments. AI had a bubble just like the dot-com bubble, so that helped in creating AI-based mechanisms for all our scientific needs as well.
Yeah.
So, is there a business aspect that will help research in India to get better? Like us pitching a very novel idea, take it on a global
for
scale.
personal DNA sequencing?
No, not just, it was actually marketed as a personal DNA sequencing. So they put a business aspect into that. So it was readily made available for a lesser cost than what was used to, we were used to 10 years back or so. So is there a business aspect to research in India, which if we pitch properly, we'll have a great development inside on attract investors?
Yeah,
So
see, that's a
that can
it's an
speed up the
interesting
process of
question.
research.
And I think as of now, I don't think there is a market for personalized
No, not
DNA
just, I'm just
sequencing.
using it as an example. So it was very costly, and only because it was marketed as a personal use device, at home device, its cost was able to come down very drastically. So
That's what. See, if in as far as India is concerned, India's sort of market demands are very different from what the rest of the world has. In the US, even right now, you can get your DNA sequenced in India also, by the way, but hardly anybody does it, right? And the reason is, I believe that yeah, it's okay to know, but what will you do with that information, right? I mean, if you are going to, is it going to help certain diseases? Yes, disease markers, it's okay, like breast
no,
cancer.
not
That's a good market if you
just
really
not
want to
to
check.
respect to DNA sequencing, with respect to all aspects of our research towards antibiotic resistance or vaccine development
Yeah,
and
I
all
mean
that.
that already exists, right? The nanopore device, the nanopore sequencer is precisely the kind of device that you are suggesting.
Okay.
It makes it really will tell you whatever, you know, in a small, it's about this big. You can connect it to your laptop,
Yeah.
and you can use it. They are using it on the International Space Station also to quickly sequence if somebody falls sick, to see what is it that's this thing and all that. And that exists. That's there already in the market. We don't use it a lot because there are better technologies out there, but you can do it. If you have
Just like a rapid test kit.
you can use it. Yeah.
Thank you.
Nothing like very small moment of weekend.
Oh, thank you, sir. Thank you. Thank you. Thank you, sir. Okay, thank you, everyone. I'll be here if you have any more questions.
Yeah, thank you so much. Thanks,
you,
thank the
sir.
audience for being part of
Thank
this lecture, and thank you so much,
Thank you.
Dr. Arun Panchapakesan. I mean, more than anything, the way you had presented, it's such ease. I was able to understand. I have no background in biology. Yeah, so and especially, I mean, the way you decoded the idea of a billion and how that means that how many cells and what parts of the cells that we have. And yes, I think we should actually spread the lecture, and those who had joined late, you can actually watch the lecture online. It's there in Popular Science Lecture YouTube channel. Yeah, thank you.
Thank you.
Excellent.
Thank you, sir.
Okay, I really appreciated the after a long time, after hearing your lecture.
Thank you, sir.
The whole thing makes sense only in the context of
Evolution.
Yes.